Hello students, welcome back to Nucleus Neet Academy and I am Kiran Kumar, your Biology Neet faculty. Today we are going to start another wonderful topic from biomolecules, that is nucleic acids. Already we have discussed the three very important biomolecules, carbohydrates, proteins and lipids. Just like these biomolecules, nucleic acids are also a kind of biomolecules which are present in the living cells of living organisms. Nucleic acids play a very important role in living cells. Now let us discuss about nucleic acids. Nucleic acids, they are acidic biomolecules. In the name itself is there, they are acids. Since they were first isolated from nucleus of pus cells, that's why the name nucleic acids. Chemically, nucleic acids, they are heteropolymers of nucleotides. What is heteropolymers? Students, hetero means different, polymers means large molecules which are formed by several repeating units called as monomers. So what are the repeating units or monomers here? Nucleotides. I will tell you what are nucleotides in a moment. Imagine that a nucleotide is just one molecule, okay? And when several nucleotides are attached to one another by bonds, it results in formation of a long chain of nucleotides just like this. Then we call it as a nucleic acid or a polynucleotide. Okay, imagine that each ball is a nucleotide. Each ball is a nucleotide. One nucleotide, it forms a bond with another nucleotide. This bond is called phosphodiester bond. Can you repeat it? Phosphodiester bond. So then it is called a dinucleotide. This molecule is called as dinucleotide. Second nucleotide, again it forms a bond with third nucleotide, again by phosphodiester bond. Now it is called a trinucleotide. Same manner, third with fourth, fourth with fifth, fifth with sixth, sixth with seventh, seventh with eighth, and so on. It results in a very long chain of nucleotides. Now it is a heteropolymer. It is made up of different types of nucleotides. This large molecule, we are calling it as a nucleic acid. Okay, we can also call it as polynucleotide also, because poly means what? Many. It is made up of many nucleotides. Understood the concept? Understood everyone? Yes. Now, what are the different types of nucleic acids? There are majorly two types of nucleic acids in living organisms. One is the DNA and another is RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. Okay. So DNA, where it is present? DNA is present inside the nucleus of eukaryotic cells, and DNA is also present in the prokaryotic cells in the cytoplasm. And RNA, ribonucleic acid, RNA is also present in both eukaryotic cells as well as prokaryotic cells. Now let us discuss the functions of DNA. DNA mainly, it acts as genetic material. It acts as a genetic material by storing genetic information. I will make you to understand very easily. If you look at ourselves, okay, we have several characters, external characters. We have our own unique facial features. We have our own skin tone. We have our own nose, our own ear. Okay. So this type of facial features we don't find in any of them. Okay. The face which you have is unique. No other person will have that same facial feature except identical twins. Okay. So all the characters in our body, such as the color of the hair, the color of eyes, the shape of the nose, the shape of the mouth, okay, the shape of the entire face, all these characters which we can see from outside, these characters we have got from our parents. Am I right? So how we get these characters, how we inherit these characters from our parents? Through genetic material. So that is nothing but your DNA, deoxyribonucleic acid. So with the help of this molecule called as DNA, the characters from our parents get transmitted to us. So this is how the DNA acts as genetic material by storing the genetic information. Suppose if you want to store some video files, some audio files, or we want to store some PDF files. How we store them? We store them by using a pen drive, or we may use a hard disk also. Okay, so here hard disk and pen drive, they are serving as aids to store the information or the data, computerized data. So living organisms, how they store the information that represent all various characters? They use DNA, that is deoxyribonucleic acid. So all the information, biological information, will be stored in this wonderful molecule called as DNA, deoxyribonucleic acid, and that's why the DNA is called as the genetic material in all the living organisms. Understood the concept? I hope you have understood very well. Next, RNA. RNA can act as different, it has multiple roles. It can take different types of responsibilities inside the cell. RNA can act as a ribozyme. It is an enzyme which shows catalytic activity, which can catalyze the chemical reaction. Our RNA can also act as an adapter. It can transfer the amino acids from cytoplasm towards the ribosomes. RNA can also act as a messenger to transfer the genetic information from DNA to proteins. RNA can also be a component of ribosome. It is one of the structural components in the ribosome, a cell organelle, and it can also act as a genetic material in some viruses. Not in all viruses. In some viruses, RNA can also act as genetic material by storing the genetic information, and there are other functions of RNA also. Okay, now let us discuss about the composition of DNA, deoxyribonucleic acid. DNA is made up of three components: nitrogenous bases, deoxyribose sugar, and phosphoric acid. Nitrogenous bases, they are basic compounds or basic molecules. They are rich in nitrogen. That's why the name nitrogenous bases. There are two types of nitrogenous bases, purines and pyrimidines. As you are seeing here, purines have double rings. There are two rings in their structure which are fused with each other. Whereas pyrimidines, there is only one ring. You can see here. Okay, there are two types of purines, adenine and guanine. Adenine is represented by capital A, guanine is represented by capital G. Pyrimidines, again two types, cytosine represented by capital C, thymine represented by capital T. A very important point, thymine is also called as 5-methyl uracil. Very important point. Okay, now look carefully here. In purine structure, if you observe, the nitrogens are occupying first, third, seventh and ninth positions. In structure of pyrimidines, the nitrogen is occupying first and third position. Got the point? Now, deoxyribose sugar, that is formula C5H10O4. Generally, ribose is a 5-carbon sugar. Here, one oxygen is less. That's why the name deoxyribose sugar, C5H10O4. It is a sugar. Phosphoric acid, H3PO4. DNA gets acidic nature because of phosphoric acid. Very important. Okay. Now, ribonucleic acid, RNA. RNA is also composed of three components: nitrogenous bases, ribose sugar, phosphoric acid. So what is the main difference between DNA and RNA with respect to sugar is, here it is deoxyribose sugar, here it is ribose sugar. Ribose sugar formula is C5H10O5. Phosphoric acid, same phosphoric acid is present, and just like DNA or RNA is also acidic in nature, that's why it is called ribonucleic acid. Now coming to the nitrogenous bases, unlike our DNA, our RNA is also having two types of nitrogenous bases, purines and pyrimidines. Purines, same again, adenine and guanine, same adenine and guanine are present. Pyrimidines, same cytosine is present, but thymine is absent. Instead of thymine, there is another nitrogenous base called uracil. Understood? So this is about the composition of DNA and RNA. So DNA is chemically more stable, but RNA is less stable, mainly because of highly reactive nitrogenous base called uracil. Okay, so RNA is less stable when compared to DNA, mainly because of what? Mainly because of highly reactive uracil nitrogenous base. Understood? So copy down this information, students. Students, now we will understand the concept of nucleoside and nucleotide. What is a nucleoside? A nucleoside is a molecule formed by fusion of ribose sugar molecule and nitrogenous base. Okay, a ribose sugar and a nitrogenous base, when two molecules fuse with each other, resultant molecule formed is called as nucleoside. Look here, this is ribose sugar structure. Ribose sugar is having 5 carbons. This is the first carbon position, second position, third carbon position, fourth carbon, fifth carbon, and there will be one oxygen atom in the structure of ribose sugar. Nitrogenous base, it can be adenine, guanine, cytosine, thymine, or even uracil also. Okay, so when any type of nitrogenous base, when it forms a bond with the first carbon of ribose sugar, the bond is called as N-glycosidic bond. N-glycosidic bond. Now, the resultant molecule so formed, it is called as a nucleoside. Okay, so that is why I wrote here a nucleoside is combination of ribose sugar and a nitrogenous base. Okay, any one of this nitrogenous base can be here. Okay, what is the bond? The bond is called as N-glycosidic bond. Very important for your NEET exam. In a nucleoside, nitrogenous base is attached to ribose sugar by bond called N-glycosidic bond. Okay, now let's come to nucleotide. What is a nucleotide? A nucleotide is a combination of phosphoric acid H3PO4 plus a ribose sugar plus a nitrogenous base. Okay, or simply you can write it is a combination of nucleoside and phosphoric acid. When a nucleoside is attached with another molecule called phosphoric acid, it results in formation of a nucleotide. Now let us look at this structure. A ribose sugar is already attached to a nitrogenous base by a bond called N-glycosidic bond. Now this is the phosphoric acid molecule H3PO4. Okay, so now elimination of water molecule takes place during bond formation. This hydrogen, this OH group get eliminated in the form of H2O, water molecule. After elimination of water molecule, a bond is formed, a new bond is formed between fifth carbon of ribose sugar and oxygen of phosphate group. This bond we are calling it as ester bond. What is the bond called? It is called as ester bond. So now the resultant molecule is called as a nucleotide. A nucleotide is acidic nature or basic in nature, can you tell? Obviously a nucleotide is always acidic in nature because of presence of acidic group called as phosphoric acid. Because of presence of phosphoric acid. And what are the types of bonds? How many types of bonds are present in ester bond? I mean, how many types of bonds are present in a nucleotide? There are 2 types of bonds. One is N-glycosidic bond and another is ester bond. And what type of bonds are present in a nucleoside? There is only one type of bond, that is called as N-glycosidic bond in a nucleoside, that will bind a nitrogenous base with a ribose sugar. Understood the concept, students? Copy this. Students, now let us represent the different types of nucleosides present in the nucleic acids. First, we will discuss about deoxyribonucleosides. Deoxyribonucleosides. So, what are deoxyribonucleosides? These are nucleosides which have deoxyribose sugar. What type of sugar? Deoxyribose sugar. Okay. So, you know what is the difference between deoxyribose sugar and just ribose sugar, right? So, deoxyribose sugar, when it is attached with adenine, nitrogenous base, then the resultant nucleoside which is formed is called adenosine, deoxyadenosine. So this is the corresponding nucleoside. Okay. Same deoxyribose sugar, when it combines with guanine, then it results in formation of deoxyguanosine. Deoxyribose sugar, when it combines with cytosine, then it gives rise to a nucleoside called deoxycytidine. A deoxyribose sugar, when it combines with thymine nitrogenous base, it gives rise to a nucleoside called deoxythymidine. So these are the four different possible deoxyribonucleosides. Okay, they will be present in DNA. Okay, now just ribonucleosides. Ribonucleosides contain just a ribose sugar and a nitrogenous base, and they will be present only in RNA, not in the DNA. Okay. So what is the sugar? Just ribose sugar. Ribose sugar plus adenine gives rise to just adenosine, which is a nucleoside. Ribose sugar plus guanine gives rise to ribonucleoside called as guanosine. Ribose sugar plus cytosine gives rise to cytidine. Ribose sugar with uracil gives rise to uridine. So here ribonucleosides, they will be present in RNA, not in DNA. So in RNA there will not be thymine, there will be uracil. So here the corresponding nucleoside is called as uridine. So these are the four nucleosides which are present in RNA, and these are the four nucleosides which are present in DNA. Okay. Understood, students? Now let us discuss about the possible nucleotides. So just like the deoxyribonucleosides, here also deoxyribonucleotides are possible. So when a nucleoside called as deoxyadenosine, it is a nucleoside. When it combines with the phosphoric acid H3PO4, then it gives rise to a molecule called as deoxyadenilic acid. So what is the nucleotide called? Deoxyadenilic acid. It can be also represented as DAMP. DAMP stands for deoxyadenosine monophosphate. Deoxyadenosine monophosphate. Okay, so same. Deoxyguanosine plus phosphoric acid gives rise to deoxyguanilic acid. Guanilic acid. Deoxyguanilic acid can be also called as DGMP, that is deoxyguanosine monophosphate. Deoxyguanosine monophosphate. Next, deoxycytidine. Deoxycytidine, when combines with phosphoric acid, gives rise to deoxycytidilic acid. This is also called as DCMP. DCMP stands for deoxycytosine cytidine monophosphate. Deoxycytidine monophosphate. Next, deoxythymidine. Deoxythymidine, when combines with phosphoric acid, it gives rise to deoxythymidilic acid. Thymidilic acid. It can be also called as DTMP. DTMP stands for deoxythymidine monophosphate. Okay, so these are the four types of nucleotides. These are the four types of nucleotides which will be present in DNA. They will be present in DNA, deoxyribonucleic acid. Then what about the corresponding ribonucleotides? Ribonucleotides. Ribonucleotides, obviously they will be present in RNA. So what are the four types of ribonucleotides possible? So already we have written here adenosine. Adenosine plus phosphoric acid, H3PO4, gives rise to just adenylate acid. Adenylate acid. You can simply write this as AMP, adenosine monophosphate. There is no D. We have removed D. Deoxy is not there. Just it is adenosine monophosphate, otherwise called as adenylate acid. Next is guanosine. Guanosine, when it combines with phosphoric acid, it forms a corresponding nucleotide called guanilic acid. Guanilic acid can be also called just GMP, guanosine monophosphate. Understood? Next, cytidine. Cytidine is a nucleoside. When it combines with phosphoric acid, then it gives rise to cytidilic acid. Cytidilic acid, simply called as CMP. Cytidilic acid. So here in ribonucleotides there will not be thymine, there will be uracil. So uridine is the corresponding nucleoside. Uridine, when it combines with phosphoric acid, it forms nucleotide called uridylic acid, simply called UMP. Uridine monophosphate. So these are the different types of nucleotides present in DNA and RNA. These are the nucleosides present in DNA and RNA. Understood the concept? Very important. Many questions will be asked in the Neet examination based upon this nucleotide and nucleoside concept. Okay, so look here, very important. Uridylic acid will be present only in RNA, not in the DNA. Uridine will be present only in RNA, not in DNA. Okay, so copy down, students. You have to memorize two structures of nitrogenous bases, very important for your NEET examination. Structure of adenine and structure of uracil. In adenine structure you can see, which is basically a purine, it is made up of two rings. First ring is attached to the second ring. First ring is a 1, 2, 3, 4, 5, 6 membered ring, and the second ring is 1, 2, 3, 4, 5 membered ring. Okay, so here two ring structures are fused with each other in structure of adenine. If you look at the composition of different members of the ring, the first ring have carbon, nitrogen, and second ring is also having carbon and nitrogen. So since in this structure of adenine there are more than one member, we call this type of ring structures as heterocyclic ring structures. Okay, so adenine shows heterocyclic ring structure because the ring has more than one member. So that is, carbon is there and also nitrogen is there. That's why it is a heterocyclic ring structure. So here nitrogen is present in first, third, 7th and 9th position of this adenine ring structure. So nitrogen occupies first, third, 7th and 9th positions. Okay, so this is about the structure of adenine, and there is NH2 group attached to the 6th carbon. A proton is present on 7th nitrogen and proton present on 9th nitrogen. Coming to the structure of uracil, which is basically a pyrimidine. Uracil shows a single ring structure. It is also composed of more than one element. Carbon is present, and along with carbon, nitrogen is also present in the ring structure. This is also a heterocyclic compound, a heterocyclic molecule. Here you can see carbon attached with oxygen by a double bond, second carbon, and fourth carbon is attached to oxygen by a double bond, and the third nitrogen is having a proton and first nitrogen is having a proton, and remaining places you can see just carbons. This is the structure of uracil, which is a pyrimidine. Okay, uracil is present in RNA, whereas adenine, it is found both in DNA and RNA, right? Next, coming to phosphodiester bond formation, how a phosphodiester bond is formed. So basically a phosphodiester bond is a bond which will fuse two nucleotides with each other. So imagine this is a nucleotide and there is another nucleotide. So a bond called as phosphodiester bond, will link the two nucleotides together. Now let us see how this phosphodiester bond is formed between two nucleotides. Okay, so here you can see this is a nucleotide, a general structure of nucleotide showing a ribose sugar, a nitrogenous base and a phosphate group. Okay, here I have represented another nucleotide. Okay, so imagine that this is a first nucleotide and this is the second nucleotide. Now when two nucleotides come together, come closer to each other, then a bond formation will take place involving removal of water molecule. Look here in this phosphate group of the second nucleotide, the OH and the proton which is present in the first nucleotide, they will be removed in the form of a water molecule. Once again I repeat, the OH from the phosphate group and a proton from the nucleotide on third carbon will be removed in the form of water molecule. So when a water molecule is removed, a water molecule is eliminated, then a bond will be formed between the third carbon of a nucleotide and the phosphate group. This bond is a ester bond. This bond is called as ester bond. But where is phosphodiester bond? Already there is a ester bond formed in a nucleotide. This is the old ester bond. Okay, this is the old ester bond. Now the two ester bonds combined with each other together together these two ester bonds, when they are combined with each other, these two bonds together is called as phosphodiester bond. It is called phosphodiester bond. So basically a phosphodiester bond will join the two nucleotides with each other, and a phosphodiester bond will have two ester bonds. One is the old ester bond which is present already within a nucleotide structure, and a new ester bond which is established when the two nucleotides get connected with each other by removal of a water molecule. Resultant molecule which is formed by fusion of two nucleotides, now we can call this entire molecule, this complete molecule, we can call it as dinucleotide. Dinucleotide. Di means two. So two nucleotides fused with each other. Now I will ask you a simple question. What is the number of ester bonds? The number of ester bonds in a dinucleotide, and what is the number of phosphodiester bonds in a dinucleotide? What is your answer? Come on, guess. How many ester bonds are present in dinucleotide? Look here, already there is one ester bond here, one more ester bond, and one more ester bond. So how many ester bonds? 1, 2 and 3. So total 3 ester bonds are present in a dinucleotide. And how many phosphodiester bonds are present in a dinucleotide? So 2 ester bonds combined together is equal to 1 phosphodiester bond. So there is only one phosphodiester bond. So we should not consider this ester bond under phosphodiester bond. Okay. So this is a separate ester bond. This is phosphodiester bond. Students, now look here. This is the first carbon position, second, third, fourth and fifth position. Now look. So the direction of phosphodiester bond formation, phosphodiester bond formation is from 5 prime towards 3 prime end. Okay. So this is another important point. Now you can copy down this. Students, now I am going to explain the formation of double helical structure of DNA. Listen carefully, it will be very interesting. Many questions will be asked in the NEET examination. Look here. This is one nucleotide, right? This is a nucleotide with a thymine nitrogenous base. And here you can see another nucleotide is present with guanine nitrogenous base. Now two nucleotides attached by a phosphodiester bond. Now third nucleotide is also attached by another phosphodiester bond. Now fourth nucleotide is also attached by phosphodiester bond. Now total how many nucleotides are there in this chain? 1, 2, 3 and 4. 4 nucleotides are present. So now we can call this as an oligonucleotide. What we can call it? Oligonucleotide chain. Oligo means what? Oligo means few. Oligo means few. Few nucleotides are present in this chain. That's why we are calling it as oligonucleotide chain. So if many nucleotides are present, like more than 10, then we can call it as polynucleotide chain. So in a polynucleotide chain or a oligonucleotide chain, there will be two ends. One is 3 prime end with free OH group and 5 prime end with the free phosphate group. Understood? So here I have represented another polynucleotide chain parallel to it, but in reverse direction. You can see I have inverted the ribose sugar. Now there is a free OH group in this end. This is called as 3 prime OH group. Here you can see 3 prime OH group, and opposite side you can see free phosphate group. This is a 5 prime phosphate. Okay, so now here you can see the two strands, this is one strand of polynucleotide chain, this is another strand of polynucleotide chain. Now both have been arranged anti-parallel to each other. So one is facing 5' to 3' direction, and another strand on other side, it is facing 3' towards 5' end. So that is anti-parallel arrangement of two polynucleotide strands. Okay, when two strands come opposite to each other, then there will be a formation of beautiful hydrogen bonds. Look here, since thymine and adenine are complementary to each other, they can form two hydrogen bonds. Thymine pairs with adenine by how many hydrogen bonds? By two hydrogen bonds. And similarly, adenine also can pair with thymine by two hydrogen bonds. Okay, so here you can see guanine, it forms three hydrogen bonds with cytosine, and here again adenine forms two hydrogen bonds with thymine. Here cytosine again it forms three hydrogen bonds with guanine. So like this, the hydrogen bonds will attach the two polynucleotides with each other. Okay, so what are these bonds? These bonds are called as hydrogen bonds. So these hydrogen bonds, they will stabilize the double helical structure of DNA. They will stabilize the structure of DNA. Okay, if we break the hydrogen bonds, then what will happen? The two polynucleotide chains will separate from each other. Understood the concept? Now look here. In a polynucleotide chain, there will be sugar and phosphate which will be arranged alternately. Okay, the sugar and phosphate will form the backbone structure of the DNA. Here you can also see sugar, phosphate, sugar, phosphate. Here also sugar, phosphate, sugar, phosphate. So sugars and phosphates will form the backbone structure of the DNA, whereas the nitrogenous bases, they will be arranged perpendicular to the backbone of the DNA. Understood? So the same structure I have represented here. So a single line represents a polynucleotide chain. So two lines represent two polynucleotide chains, and this is the 5' end, and the opposite end, this is the 3' end. Okay, then automatically this end becomes 3' end and this end becomes 5' end of polynucleotide chain. Okay, so here on a polynucleotide chain you can find the sugar and phosphate backbone. So here I am representing sugars, sugar, sugar. So between two adjacent sugar, there will be a phosphate group. There will be a phosphate group like this. There will be a phosphate group. So this is what we call it as a sugar phosphate backbone. Okay, so there will be two sugar phosphate backbones in a double helical structure of DNA. Okay, now look here. Wherever sugars are present, wherever sugars will be present, attached to sugar there will be also nitrogenous base. If adenine is present on opposite sugar, there will be thymine. Adenine and thymine will always pair with each other and they form double hydrogen bonds. Okay, suppose here in a sugar phosphate backbone, here in this sugar there is a guanine, and opposite sugar there will be cytosine. In opposite sugar there will be cytosine. Guanine and cytosine will form triple hydrogen bond. Understood? So imagine that here another sugar is having thymine, then the opposite sugar would have adenine, and adenine thymine would pair with again by double hydrogen bond. Suppose here the sugar has cytosine, then the opposite sugar will have guanine and they will be held by three hydrogen bonds. So like this, the base pairing will take place between the two polynucleotide chains. Okay, So this structure which you are seeing here, it is called as a double helical structure of DNA. Double helical structure of DNA given by two scientists, Watson and Crick. Watson and Crick. So the two strands of polynucleotides, they are twisted like a helix in a right hand direction, and that gives the appearance of a double helical structure. Okay, so here Watson and Crick, they have proposed this double helical structure based upon X-ray crystallography data. So according to the double helical structure proposed by the two scientists Watson and Crick, the two strands of DNA are arranged anti-parallel to each other. One strand is arranged 5' to 3' direction, whereas opposite strand is in 3' to 5' direction. Okay, so one strand is facing 5' to 3', another strand is facing 3' to 5'. So this type of arrangement is called anti-parallel arrangement. It is called anti-parallel arrangement of DNA. Okay, so here the sugar and phosphates will form the backbone of the entire DNA structure on both the strands. So here I have not represented the phosphate groups, but you have to imagine that there will be phosphates here. Okay, just like this. Since it becomes more complicated if I draw phosphates, that's why I didn't represent phosphates here. Okay, so that is the thing. So now how the base pairs are arranged? Guanine and cytosine, this is one base pair. Thymine and adenine, this is another base pair. Cytosine and guanine is another base pair. So each of these two nitrogenous bases which are held by hydrogen bonds, they are called as base pairs. Understood? The base pairs are arranged perpendicular. They are arranged perpendicular to the sugar phosphate backbone towards inside. Okay, so now you look here, the DNA, it takes right hand turn. It takes a right hand turn. When the DNA is taking a right hand turn, it has to cover a distance. It has to cover a distance, complete distance of 34 Armstrongs. 34 Armstrongs in order to complete one complete turn of 360 degrees. In order to complete one complete turn of 360 degrees, the DNA has to cover a distance of 34 Armstrongs. Okay, so this much distance the DNA can complete one 360 degrees turn. So one turn of 360 degrees requires 34 Armstrongs distance, and it is called the pitch of the DNA, otherwise called as the gyre of the DNA. So 34 Armstrongs represents pitch of the DNA, otherwise called as gyre of DNA. So this is the distance which is needed for DNA to take one complete 360 degrees complete turn. Understood? So now one complete turn has 10 base pairs. So from here to here, if you estimate the number of base pairs, then approximately it will be about 10 base pairs. Okay, from here to here. Let us count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So there will be 10 base pairs in this distance of 34 Armstrongs. That is what I have written here. One complete turn has 10 base pairs. Okay, now angle at each step of ascent is 36 degrees. So this is, if you imagine a base pair as one step and another adjacent base pair as another step, so the angle at which the two adjacent base pairs are arranged, that angle will be 36 degrees. It is called as angle at each step of ascent. Otherwise you can also call as the distance between adjacent, or you can also call as the angle between the adjacent base pairs. That angle is going to be 36 degrees. Okay, then the distance between adjacent base pairs, this distance will be 3.4 Armstrongs. The distance between any two adjacent base pairs, it will be 3.4 Armstrongs. Here I am writing distance dt, distance between adjacent base pairs. BP means base pairs is how much? 3.4 Armstrongs. 3.4 Armstrongs. Okay. So the distance between base pair can be also called as the rise per base pair. Rise per base pair means how much distance the DNA is increasing in height for every base pair. So the rise per base pair is also 3.4 Armstrongs. It is nothing but distance between adjacent base pairs. Next, width of DNA. So what is the width of DNA? The width of DNA was found to be 20 Armstrongs. The width of DNA was found to be 20 Armstrongs by Watson and Crick. Okay, so this is about the double helical structure of DNA as proposed by Watson and Crick. So whenever a DNA is having 10 base pairs, whenever DNA is having 10 base pairs per one complete turn, that DNA is called bDNA. So bDNA, what is the character of bDNA? bDNA has 10 base pairs per turn. Okay, so in higher classes, you will be knowing about many other types of DNAs that will be differing from each other based upon the number of base pairs in each turn. Okay, there is also aDNA, cDNA, dDNA, zDNA. So many types of DNAs are present, but the most common type of DNA is bDNA, which is having 10 base pairs per turn. And our Watson and Crick scientists, they have proposed the structure of bDNA. Understood? Copy down this, students.